I'm David Christini. I'm the Dean of the School of Graduate Studies, and welcome to the keynote session for our annual Research Day. One of our graduate students, Zhao Mariah, is going to do the introduction in a moment of our keynote speaker. I'm just here to welcome you. I think it's been a really exciting day so far. We started off with some platform talks from our excellent graduate students, followed by a lunch, and then some fantastic poster presentations. I think that for me personally, this is always a really fun event, seeing exciting new science and eager developing scientists. I think this year in particular, it's especially, it feels that way especially. I think we've all heard people describe the current system the current situation out of the federal government. Specifically, I'll keep my comments specifically related to science and higher education funding being described as unprecedented. It clearly is unprecedented, but in my mind, that word just doesn't really do justice. I think those of us who have been in science for a while have seen a number of things over the years, every decade or so, that are unprecedented, some bad, some good. But I would go further to say, frankly, what's happening right now is unimaginable. I think to many of us, it is just so bad what's happening to science. But that's why today is particularly refreshing, because we can keep our eye focused on what we can control, and what we can control is the work that we do here at Downstate, our faculty, our postdocs, our graduate students, our research support team, and that our colleagues do across the country, such as at Vanderbilt, as we're going to hear in a moment, and just keep trying to demonstrate the importance and value of science and what it contributes to society and health. And so that's one of the things that I personally am trying to keep focus on right now, is doing what we can do to try to keep pushing things forward. So thank you to all of you for doing everything that you're doing. I didn't mean that to sound somber. I hope it didn't. It's not a great time we're in, but I am optimistic locally in what we're able to do. And with that, I'm going to hand the microphone over to Joao for the introduction.
So, hello everyone. It's my pleasure to introduce today's speakers, Dr. Suzana Herculano-Houzel. Dr. Herculano-Houzel is a renowned Brazilian neuroscientist, widely recognized for her groundbreaking work on neuroanatomy and brain evolution. She became famous for refining a method that precisely estimates the number of neurons in the brain of various species, ranging from humans to dinosaurs. Currently, she serves as an associate professor of psychology at Vanderbilt University, where her research explores how neuron count affects cognitive capacity, longevity, sleep, and human evolution. She also investigates how cortical folding follows the laws of physics and studies how our brain manages their development in an environment with limited energy supply. She's also the first female editor-in-chief of the Journal of Comparative Neurology and a columnist at the Brazilian newspaper Folha de São Paulo. Her online talks have attracted millions of viewers, showcasing her talent not only as a top-tier researcher, but also as an excellent science communicator. She's the author of The Human Advantage, a new understanding of how our brains became remarkable, where she explains that humans stand out not because of our brain size, but because we have more neurons in the cerebral cortex than any other animal, thanks largely to our ancestors' invention of cooking. The topic of her talk today will be From a Demand-Based to a Supply-Limited View of Brain Development, Function, and Evolution. Please join me in welcoming Dr. Suzana Herculano-Houzel.
Thank you so much for the invitation to be here and speak to your young scientists. I love talking about science. I especially love talking about my own research, but most of all, I love talking to young scientists who have all sorts of possibilities before them. And also because young scientists haven't become tainted yet by all the intuitions and of course it's this way that we get very often spoon-fed and led to believe when sometimes we actually don't know the first thing about what we're looking at, which is definitely the background to the story that I'm going to tell you today. It all started with my interest in understanding the diversity of brains and the bodies around those brains. What is the extent of that diversity? What are the constraints to that diversity? And even more than that, so what that you have all this diversity? Let me give you a snapshot of what this diversity in brain size and body size that I'm talking about is. You're looking at each data point here in each of these graphs is one species. In this case here, different species of birds, different species of mammals, or both birds and mammals here at the bottom where I'm showing you birds in black. You may glimpse some overall trends, like for instance, the bigger an animal is, the generally longer that it takes to gestate a new animal, although there's no universal relationship there. The bigger the relative size of the brain is, as in what percentage of the body does the brain represent. The bigger that relative brain size is, the fewer the neurons you have in the cortex, though again, no real unity there in either birds or mammals, and no universal relationship between how much does each gram of body use, what's the rate of use of energy per each gram of body, and how many neurons you have in the cortex of that species, which altogether means that, have been the arguments why people would say that, well, see, all this diversity must arise on a need basis through adaptation by way of the survival of the fittest, which means that some of these animals must have been selected for this or for that, and that would be the reason or the origin of all this diversity that doesn't get captured by any one universal scaling rule. I am, of course, in the business of understanding whether there actually are universal scaling rules, and I will, of course, propose to you that actually, yeah, there is actually a very simple scaling rule that ties all these variables together in what turns out to be one very simple equation, so simple that it's the kind of equation that I hope by the time I'm done here, you're all going to be saying, well, duh, of course, but that's where you see the huge gap that lies in between what we've been told, what we've been taught to think and expect that every single animal is adapted through natural selection for what it needs to have. So this is what I'm calling here the demand-based view of how animals and brains and neurons work, what they look like and how they function. Presumably, this is what we've been taught to expect, that animals need something, just like brains need something and neurons need something. What I will propose to you is that there's another way of thinking, of looking at the same data, which is a supply-based view of not what animals' brains or neurons need, but rather what do they get. And this comes from an economic point of view, where all that matters really is, do you function, do you operate in a state where there or ample resources, where resources are never limiting, so you have the privilege of just going with whatever is best? Or do you live in a situation, do you function in a situation where resources are always limiting? Where this starts is, you've probably, even if you haven't seen this graph, you know of this graph, because this is the 1973 picture that Harry Jerrison used to explain what made humans special, which was the fact that we are the species, each data point here is 1 mammalian species. We're the species that stands out the most, that stands the farthest from this line here that you see that relates the size of a brain to the size of a body. So just look at this graph, and in plain English, you could express this graph as, well, the bigger a mammal is, the bigger its brain is. Why would that be the case? The Jerrison's expectation, along with most people, would be that, well, this line arises from the need of larger animals to have larger brains operated with more neurons, because a larger body must require more neurons to operate that body, right? Makes intuitive sense in a way, at least to a biologist, definitely to a neuroscientist, certainly to Jerrison and all the people who bought into his idea that, well, if a larger body needs a bigger brain with more neurons, and humans deviate so much, we have such a bigger brain than you would expect the brain of a generic mammal of our body size to have, that must be the root source of our exceptionality. So this is where it stands, where it all comes from, this intuition that a larger body should require a larger brain with more neurons to operate it. Now, the interesting thing is, pose the very same question to an engineer, and they're gonna say, what? No way! You can operate a tiny little robot and a gigantic robot with the exact same number of processing units, provided that the number of joints, the degrees of freedom of movement are still the same. So no, you don't need more neurons to operate a bigger body. Well, what do the data actually say? So to Jerrison's credit, he was doing the best that he could with the data that was available at the time. In 1973, we had no idea how many neurons different brains were made of, so you couldn't look at that directly. As João mentioned, 20 years ago, I came up with this pretty much insane idea of let's just take a fixed brain and turn it into soup so that we can count how many cells it's made of and how many of those cells are neurons, and that should give us direct numbers of, direct estimates of the numbers of neurons that form different parts of the brain. Like, for instance, what I'm showing here on the y-axis, let's call this the number of neurons in the brainstem regions that operate the body directly. Each data point here that you're looking at is one species whose brains have been turned into soup. So we have these numbers directly, and you're looking at those numbers of neurons in the regions that directly operate the body, plotted as a function of the size of the body of each of those species. And I hope that it's immediately apparent to you that even though there is an overall trend here, you see that the same number of neurons, just pull a horizontal line here, and you see that bodies very small and very large can be operated by the same number of neurons in the brainstem. And these are all functional animals, so you really cannot make an argument that any one works better or worse than the others. Pull a vertical line, and you see that you can have animals of a similar body mass, say about 1 kilogram, their bodies are operated by numbers of neurons that vary by a factor of up to 500. Again, you can't really make an argument that some animals are more capable than others. So what you have here is really no evidence in support of the idea that a larger body needs a larger number of neurons to operate it. Instead of looking at the number of neurons in the brainstem regions, let's look at the number of neurons in the cortex that give you associative functions, cognitive capabilities. Again, no relationship, no universal relationship that applies across all different types of animals. For a similar body mass, you find many more cortical neurons in songbirds and parrots than you find in most primates, which in turn have more cortical neurons than you find in other non-primate mammals of a similar body mass, which in turn have plenty more cortical neurons than reptiles of a similar body mass. Move on to just looking at the size of the brain of different species, and you see that, once more, there's a universal trend. The larger the body, the relatively smaller the brain. What this means is that larger bodies do have larger brains, but a body that is 10 times larger does not have a 10 times larger brain. It has maybe a 4 or 5 times larger brain, probably actually much less than that. So what we have here is evidence that there are very different ways. There's a lot of diversity, apparently, in how nature puts a brain into a body and then neurons into that brain. So I like to think that that is very good evidence against this idea that brains and bodies are built on a need basis, that they're optimized for something. Now, if you look at what down to the bare bones of this diversity, there are 2 things that are absolute requirements for life. One is structural integrity. The second one is energy. And, well, we can take structural integrity for granted here since all of these creatures exist. But let's look at so this is what I'm going to be doing with you for the rest of my talk, is let's look at how this picture comes together if we take the standpoint of energy supply, that the one thing that animals that in bodies and brains truly need is energy. Then the question that I pose to you is on a demand-based scenario, a demand-based economy, you would have, you are interested in the question of how much energy do brains use, and you would assume that animals get in brains and neurons, they get energy as needed from a supply that is, for practical purposes, unlimited, or at least not limiting. Now, on a supply-based economy, so what I'm saying here is if brains and bodies functioned as not a demand-based economy, but as a supply-based economy, then what you would expect is that how much energy a brain uses is exactly how much energy it gets. You get this much, you use that much. That's what defines a supply-limited economy. So if we want to, this is the question that I got interested in. I wanted to address first, how much energy do different brains use? What defines that? Is that the size of the brain? Is that the size of the body? Is that the number of neurons? What defines how much energy a brain uses? And then second, does that tell us something? Does that inform us about whether brains are demand-based economies or supply-based economies? How do you address that? Well, ideally, you would like to have direct data, direct information on the rate of energy use that you could pitch against the rate of energy supplied to the brain. The problem is that to do that, you need to bring live animals to the lab, whether you use radiography, 2DG, FDG, imaging, or calibrated fMRI measurements, you still have to bring live animals to the lab. We can do that with humans. We can do that with mice and rats, maybe a monkey here and there, but that's not going to happen for the wide variety of species that we need. What we can do, however, is estimate the rate of energy supplied to the brain from looking at the density of capillaries that you find in different parts of the brain and in one species and also in the brains of different species. And the reason for that is that the capillaries are the ultimate part of the vascular system, which is a distribution network, where diffusion is actually effective, where diffusion of gases between tissue and the blood effectively happens. And any and all oxygenation of the brain and transport of glucose happens through the capillaries. Now, very importantly, before somebody starts thinking, like I had to hear from uninformed reviewers, oh, but that tells you nothing, because look at muscles. We can recruit capillaries. You can have capillaries that are empty, they're closed, and on an as needed, you get those capillaries filled up. That's called capillary recruitment. That is known to not exist in the brain. Which is one very important hint already that the brain is using at all times all the energy that it can get. Virtually every single capillary in the brain is open, it's patent, there's blood flowing through it at all times. We also know that if there's any room for modifications in the density of capillaries in the brain according to any need, according to on a demand basis, those adjustments would have happened during development. So if you look at the density of capillaries in the adult brain, you're looking at the steady state, you're looking at that end state where the density of capillaries is a direct indication of the rate at which energy can be supplied to that tissue. Okay, so how does that inform us about this question of whether brain economy is demand-based or supply-constrained? Well, think of your brain as a country where the energy-guzzling units are distributed heterogeneously throughout the country, like the population on the territory. So if these are the units that use energy, you would expect that a system of delivery of goods, a system that delivers resources, and ultimately those resources have to be delivered through capillaries. If that system was built on a demand basis, then you should find that the density of capillaries in the brain should follow the distribution of the units that use that energy, exactly like the distribution of the highways in the US matches the distribution of population density in the country. For the very simple reason in this case that highways are built exactly to meet the demands of the people who live in different parts of the country. So that's the signature of a demand-based supply system. Now, on the other extreme, a supply-limited system, if the brain were a supply-limited system, then the distribution of capillaries in the brain should look exactly like the distribution of the hydrographical basin in the US. Where the rivers are could not care less about where the people are. It is what it is. So this will be part one of my talk. Does the capillary supply in the brain follow the presumed demand? which tells us, can brain energetics be demand-based, as we have been taught to just assume? Because neurons are these energy-hungry cells. They demand energy, and they get all the energy that they demand. So wherever you have more neurons, you would expect that there should be more capillaries. So I'm going to use this image here to illustrate how we went about measuring local densities of neurons that you see in red, labeled with nu-n, and pitch that against the density of the endothelial cells. So not capillaries themselves, but the individual endothelial cells that make the capillary. So one capillary is one single endothelial cell curved around itself, which means that every capillary is exactly one endothelial cell thick. Which means that when we measure the local density of endothelial cells, we have a proxy for the density of capillary length in that tissue. So the work that I'll show you for now was the work of a graduate student in the lab, Lisa Ventura Antunes, who's now a postdoc at Vanderbilt. And the question that Lisa asked was essentially, well, we have clear predictions that we can make, opposing predictions on one and the other scenario. So if on a neuron-centered demand-based economy where neurons require energy, you should see that you would expect that the rate at which energy is required should depend on maybe it's the number of synapses, maybe it's the number of neurons, maybe it's the surface of neurons, but in any of these cases, which we're not measuring, to be clear, you would expect that local capillary densities should vary together with the local densities of neurons, which we know to be extremely variable across sites in the brain. So that's the DE, the density of endothelial cells, that should vary together with the local density of neurons. In a supply-limited scenario, however, you should find that what determines how much energy is available for neurons to use is simply a function of capillary density there. So in this case, we would predict that you have a constant capillary density across different sites of the brain, regardless of how many neurons you find site by site. And we know that this is informative. We know that capillary density is informative of actual rates of energy supply, because there's very few data out there, but there are essentially 2 studies on rats that show that where they looked at the histology of the density of capillaries, the local densities of capillaries, and they matched that directly to radiography that showed the rate of blood flow or the rate of energy of glucose use, and you find this very, very nice linear relationship. Hence the question, does the density of capillaries accompany the density of neurons? So let's first just run our local checks that what we're measuring is really informative. What you're looking at here is in both mouse and rat, you see that there is a very direct and fairly strong relationship between the local density of endothelial cells and what we call the vascular fraction of the tissue, which is essentially of the entire surface or volume, it doesn't matter, of that section, that region of the brain, what fraction is occupied by the capillaries. And you see that it's mostly a very small fraction. It's about 3 to 4% of the volume of the different parts of the brain is occupied by capillaries. So we have the relationship that we would expect, which allows us to move on to the next question, which is the one that matters. Do you find variation, that local densities of endothelial cells vary together with local densities of neurons? This is just matching our measurements, our local measurements of endothelial cell density to local measurements of both blood flow and glucose use, both in rats. Those are the few studies available on the literature. So, which means we're in a good place here to make inferences from what density of endothelial cells we measured. That local density of endothelial cells does reflect measured rates of local blood flow and glucose use in different parts of the rat brain. So, to come back to our main question, is this any relationship to the distribution of the neurons? And the answer, I think you can see in these graphs, is essentially you have two flat lines. So the answer is no. The density of endothelial cells in different structures of the mouse brain and in different structures of the rat brain, there's some variation, of course, but that variation has nothing to do whatsoever with the local density of neurons in the different parts of the brain. So we can, if numbers of neurons, local numbers of neurons are varying enormously and the local densities of the capillary cells that supply those neurons are not varying, then we can look at a new metric that becomes very informative, which is the ratio of endothelial cells per neuron, which essentially serves as a proxy of how many capillaries do you have serving, on average, every neuron in that part of the brain. And the consequence is, because neuronal densities vary a lot, but the endothelial cell density varies very little, what you have is these negative correlations where you have larger densities, higher densities of neurons. So what this means is more smaller neurons, so sites in the brain with more and smaller neurons, like, for instance, the dentate gyres of the hippocampus, the granule cell layer of the cerebellum. Where you have more neurons, more and small neurons crammed together, the individual neuron has fewer and fewer endothelial cells providing it with energy, which means that, and conversely, in those sites where you have fewer and fewer neurons, which are also larger neurons, given that the supply is the same, you actually have fewer neurons sharing that same energy supply, which means that the average neuron gets more energy. So the picture that emerges here is one where there's only so much blood that is supplied to different parts of the brain, and that supply has to be shared by however many neurons you have in that part of the brain. Which means that once you put that together with the data available on blood flow to different parts of the brain, what you get is that where you have fewer neurons locally, each neuron gets more blood flow serving that neuron individually, and that estimated rate of blood flow per neuron follows exactly that increasing ratio between endothelial cells and neurons that we measured. The same thing applies to the actual, let me emphasize something here. So blood flow per neuron, this is supply. This is the supply end of things. So what you get here is that there's more blood supplied per neuron where there are fewer neurons in the brain. The same thing is true for the rate of actual glucose use, where in those parts of the brain where you have fewer neurons and therefore more endothelial cells per neuron, you have higher rates of glucose used per neuron on average. If we match that to densities of synapses in different parts of the mouse brain, also published data that we match to the regions where we do the analysis, you find that there's also no relationship there. So we can very, very safely exclude the initial possibility that so the data do not at all support this demand-based view of brain economy. So this is what we would have expected to see according to the traditional intuition that the brain demands energy and gets what energy it needs. Rather, what we have is evidence in favor of a supply-limited economy of the brain, where the rate of energy that is supplied, it just is what it is, and however many neurons get that energy, they must just make do with what they get. How does that translate to the function, to the actual function of a living brain? So working together with Doug Rothman, who is an expert in brain energetics and actual brain metabolism, what we worked out is that the rate of energy of use, both at rest and during activated states, say when you're engaged in a task, both activity at rest, both metabolism, metabolic rate at rest and during activation, they are fully predicted by the local densities of capillaries. So with the bottom line here, actually, that we can estimate that the adult brain, whether it's a rat or a human brain or a monkey brain, the adult brain functions at capacity. What are the consequences of that? Well, the first one we know in practice already. Stand up too fast. Don't give your circulation a chance to maintain, to change blood pressure and maintain the rate of blood flow to that constant rate of blood flow to your brain, and your sight blackens out. That's how badly we need every last drop of blood that circulates through the brain. Another consequence of that, we can make predictions about what parts of the brain should be the ones that are the most vulnerable to any kind of insult that decreases the rate of energy supply to the brain. Be it through aging, be it through ischemia, be it through metabolic diseases, just like the parts of the country where you have the highest population densities competing for limited resources, you would expect that exactly those parts of the brain that have the highest densities of neurons competing, being forced to share the energy that they're supplied with, those should be the parts of the brain that are the most vulnerable to aging, ischemia, and any kind of metabolic disease. Guess what parts of the brain those are with the highest densities of neurons? It's the hippocampus and it's the cerebellum, the granule portion of the cerebellum, along with entorhinal cortex. All of those structures exquisitely vulnerable to ischemia and to metabolic disease. One other functional consequence that can be expected from realizing that brain economy is limited by its supply to the point where every last bit of oxygen that the brain can extract from the blood it gets served with gets used, is that given that there's only so much blood circulating to the brain, through the brain, which is a known fact, that means that any part of the brain that starts using a little bit more energy, and it can be as little as just 2% or a 5% increase in local energy use, that will happen at the cost of driving flow away from other parts of the brain. Maybe that is the simplest explanation for how come we can only dedicate heightened processing to one set of functions at a time, to one functional network at a time, which also goes by the name of attention. Maybe the fact that we can only attend to one thing at a time, meaning give dedicated processing to to a set of events is limited to one thing at a time as a simple consequence of the fact that our brain, any adult brain, is already doing everything that it can with what energy it gets. So a little bit more over here is doable for some time, but only at the expense of driving it away from somewhere else. All right. Part 2. And we're getting there's a part 3 after this, but we're getting shorter and shorter here. So the question is, okay, how does it get to that point where capillary density is unchanging? How do you get capillary density to be the same across different parts of the brain? Actually, it's worse than that. This is unpublished data, again, work from Lisa, that you're looking at capillary density in the cortex, in different parts of the cortex of different species. And if you look, there we have bats and rats and pigs and a giraffe and an elephant and a raccoon and a monkey. And you see that the density of endothelial cells, the density of capillary cells, it does not change systematically with the size of the animal, with the number of neurons, with the density of neurons. No, it's like capillary density is what it is in all these different species. How do we account for that? Traditional argument is it's optimized. Every single animal gets its capillary density optimized in evolution by natural selection. I say there's a much simpler explanation for that, which is just acknowledge that the adult brain is the result of development. And we know the things that happen in development, and one of them is neuronal cell growth. So suppose that brains start out in development. At some point, there's a maximum capillary density, maximum density of capillaries formed in that brain, and your newborn neurons are intermingled there. What happens next is, on the one hand, the brain stops forming new capillaries, and there's evidence that that is the case in early postnatal development already. So you have a very high density of capillaries, and you're not getting any more new capillaries. The brain is newborn and very small. On the other hand, you have little neurons getting lots of energy. These neurons get so much energy that they can afford to grow by pushing dendrites and axons, and they grow and grow and grow, and you start getting the non-neuronal cells coming into the tissue. As they grow, as the neurons grow and the tissue grows, those capillaries are getting separated. Now, remember that the whole function of the brain is possible through the diffusion of gases between tissue and capillaries. So once those capillaries become too far apart, you've met the limit of effective diffusion that's called the Krog radius. So once the Krog radius, which is this radius separating capillaries, once you reach that limit, there is not enough energy getting to the neurons anymore that could support growth. What is the inevitable consequence is that you have a brain that has stopped growing. So that is what sets the size that a brain can have. So let me give you a quick visual of how this could work across different species. Let's say that this is just a quick and dirty representation of the density of capillaries that you have, and you expand. This brain is going to expand until it can expand no more because it's reached that Krog radius of separation between capillaries. Well, if you start from a starting point where you have a higher density of capillaries, you can predict what's going to happen to the size of this brain, right? Compared to the first one, if you're starting out from a higher density of capillaries, then when the Krog limit is reached, you have a much bigger brain. Make that starting maximal density of capillaries even larger, and you're going to be able to separate those capillaries to the same extent, but the result will be a much larger brain. So this is what I'm calling proposition number 1 here, that brain growth is a self-limiting process. Brains grow until they can grow no more, for the simple reason that brain growth amounts to neurons expanding and glial cells being added into a capillary bed that has already been completed. Finally, so what? And this is what I want to propose to you today. This is unpublished work. So what I want to propose is that not only brain size is self-limiting, but body size is also self-limiting as a consequence of that. So I'll walk you through the logic of the argument here, but so let me add to one more element. I just told you that the size of the brain depends on that maximal density of capillaries that it had. Notice that at no point I used the word number of neurons. The number of neurons does come importantly on in another way, which is my proposition number 2 here, which is the finding that I published years ago that the rate of total energy use by a brain is a linear function of how many neurons that brain has. So if you have 10 times more neurons in a brain, that brain uses 10 times more energy. Doesn't matter how large that brain is. So that's what we have here. The rate of energy used by the brain is a linear function of the number of neurons in that brain. Now, if we consider that, on the other hand, the size of the brain is determined separately, like I just told you, then we can calculate the ratio between these two things, which is how much energy each gram of brain tissue uses in the adult, right? Which is going to be proportional to the ratio between the number of neurons and the size of the brain. So this here is the specific metabolic rate of the brain, and this is the neuronal density of the brain. Now, where this becomes really important is once you realize that in every single mammal, the brain finishes growing first, which is what you see here. And so by age 5 or so, the brain has already reached about 90% of its final adult size. But the brain, the rest of the body keeps growing after the brain has finished, has reached that maximum size that it can. Why is this so important? It is because once brain growth is done, you can calculate that the average gram of brain tissue uses a certain amount of energy that is proportional to that ratio between the number of neurons and the size of the brain. So the specific metabolic rate of the brain, of the adult brain, is proportional to the density of neurons in the brain. Why is this so important? Because this specific metabolic rate, think of it as the rate at which energy is supplied from the heart to every gram of tissue in the body. Now, we know that as the body grows and the heart grows along with it, the problem is that cardiac capacity does not grow linearly with the body. So the rate at which the heart can provide each gram of tissue in the body, brain included, with energy, it goes down as the body grows. You see where this is going to hit a wall? If we know how, at the adult stage, we know how the adult brain is using every last bit of energy and oxygen that it can extract from the blood. We know that any less than that, and you risk losing consciousness. Now, as the body grows, the body is providing all parts of that body, brain included, with less and less energy per gram of tissue. So the final proposition here is that the growth of the body is limited to the size that is the maximum body size that still provides the brain with that specific metabolic rate, with that much energy per gram of brain tissue. So we can tie all of this together, and that takes us to a very simple equation that we can then test to see if it applies. So it goes like this. I told you that a capillary bed gets stretched to capacity, and that's what defines the size of the brain. Separately, you have the number of neurons that are established in some other way I'm not getting into right now, but the important part is that is what defines how much energy the brain as a whole uses. Put those two things together, and you have, for that in particular species, you have the rate of the specific metabolic rate of the brain, how much energy a gram of brain tissue costs. On the other hand, now, you have the body growing around that with a specific metabolic rate that decreases as the body grows. A fraction of that cardiac output is dedicated to the brain, presumably from early in development. That's this fraction K, which means that if by definition, if the proposition here is that this ratio here, so the body, as the body grows, this ratio drops. This ratio can only drop so far as it meets this ratio here. So this is the predicted, this right here is the predicted end state that should apply to any warm-blooded species if it's true that the size of the body is the maximum size that still provides the brain with the energy that it uses. Now, how do we test this? Let's make a couple of, we don't have, like I mentioned, we don't have measurements, direct measurements for the rate of energy used by the brain, but the theory is exactly that, well, this is a linear function of how many neurons you have in the brain or in the cortex. If we then swap these two variables here, what we have is an equation that pitches against each other. On the one hand, the relative size of the brain, so how large the brain is divided by how large the body is, multiplied by that fraction of cardiac output that goes to the brain. And on the other side, we have this ratio between how much energy the brain uses and how much energy the body as a whole uses. Now, this is useful because that gives us relative brain size as a predicted or predictable consequence of this self-limiting growth of the body to the limit at which it can, any more than that, and it can no longer feed the brain. So we can test that, and if we just plot, these are the same data that I showed you before, by the way. If we just plot the relative brain size as a function of that ratio, this ratio here between number of neurons in the brain and the metabolic rate of the body, you see what is already a decent fit, although if you pay attention, you see that primates are below the line, and so are many of the bird species. Now, this is before correcting for K. I don't have time to get into how you can calculate K. I'll be happy to tell you how we can do that. There's two different ways to calculate that K, short of actually measuring output. But the prediction is that if all of the conditions are true, if the rate of energy used by the brain is a linear function of how many neurons it has, and the size of the body is the maximum size that still supports, that still provides the brain with energy, then what we should have here is an equality that is met and that is also linear. And of course, I'm standing here showing you all this because it is, you do get this beautiful linear relationship here with a very nice fit that shows you that 65% of biological variation in the relative size of the brain and the ratio between the size of the brain of a species and the size of its body is predictable, is accounted for by this ratio between how much energy the brain is predicted to use and how much energy the body as a whole uses. So this is a very satisfying, very simple equation that can actually account for evolutionary diversity by a very simple self-limiting mechanism that determines body size to match brain size. So let me just wrap this all up for you. Take-home messages. I've shown you that we have reason to believe that the brain functions as a supply-limiting and supply-limited economy, and it functions near capacity, so it's using as much energy as it can get. The evidence for that is that the energy supplied, the rate of local energy supply depends on capillary density, which is does not match variations in local neuronal density. I've proposed to you that let's say that fairly constant capillary density that is entirely agnostic, I would go further and say that is oblivious to local densities of neurons, is the result of a self-limiting process of brain growth to the limit that the capillary bed can be stretched. Consequences of that is that heightened dedicated processing that we call attention is by force limited to one network at a time, because any increases here come at the physical cost of the redistribution of blood in the network, so more here means less that. Regions with higher neuronal densities are more vulnerable to diminished supply, and all of that because the body grows to the limit that it can still feed the brain. So you end up with findings like the human brain is powered by 22 watts from a body that functions as a whole at 70 watts. That seems extraordinary, that seems out of place compared to every other species. That is something that one of many arguments that we've been used to say that the human brain is special, it's different from everybody else, we've been selected for this and that. No, we're just one more point on the curve. Actually, we're right there on the line, on the prediction that I showed you. Because the body grows to the limit that it can still feed the brain, so what that means is that every animal has the body size that it can have and still support the brain. So put this together, and what you have is what I'm proposing as a self-limiting theory of brain and body growth and evolution, which is that animal brains and bodies are self-organized by self-limiting growth, which means that animals don't need anything. Animals get a certain amount of stuff. They're dealt a hand that specifies how many neurons you have, what's that maximum capillary density that will populate that brain, and from there, you can predict how large that brain will be, how much energy it uses, and how large is the body around it. All of it whilst doing what they can with what they get, which is the definition of a supply-limiting economy with all the interesting consequences that it has. So I'm going to wrap up here with a big thanks to all the people involved in this work, and following up on our introduction, let me make one quick comment on the so what that we humans have, as João mentioned. What distinguishes us from all the other species is that we are the species that has the most neurons in the cerebral cortex, even though everything else that I showed you is exactly what you would predict. We're not extraordinary in that sense. We are, however, remarkable in having all those neurons in the cortex because of what those neurons get us. On the one hand, unmatched cognitive capabilities, and on the other hand, lots of time that no other animal has, time that is proportional to the number of neurons, because with more neurons in the cortex, we have longer childhoods, we have a longer life. With those two things together, what we get, the main consequence of having lots of neurons in our cerebral cortex is that we have the capability of using those neurons to build wonderful things, and we have the opportunity to learn those things from the people who came before us, because we have the time to overlap as children with the generations that came before us. And that is why, to me, there is nothing that should be more sacred as a place of generation and even more than that, transfer of knowledge than a school and a university, which are by definition the places where generations come together and overlap, and we have the opportunity to use this time and this cognitive capacity that our cortical neurons give us to generate knowledge and pass it on to the next generations. That is, to me, what universities are about, and I'm going to do everything that I can to make sure that that keeps happening and that doesn't get touched. Thank you so much for your attention.
Thank you.
Do we have a little bit of time?
What's that?
No, I'm just saying, I know we're over time, but I'll
Oh, yeah. We're going to take some questions. I'm sure there's going to be some really good ones. Thank you for that really interesting talk. And if only everybody in this country agreed with your sentiments about the values of universities, I certainly do. And I think you're preaching to the choir, so to speak, here. So let's open it up to questions from the audience. If you have a question, I will bring a microphone to you. If somebody wants to, if there's any questions over there, we have another microphone. Szymantek.
Hey, so you mentioned about the supply-based thing, the model that you predicted, the equation holding up for warm-blooded animals. What would be the case for cold-blooded animals? And as far as I know, that there was a switch from being cold-blooded to warm-blooded approximately during the Triassic era, and how would that impact the evolution of the brain?
So the interesting thing about cold-blooded animals is that the rate of energy used by the brain is predicted to be not only proportional to the number of neurons, but also to the temperature in which they function, right? The other, because it's so depending on temperature, compared to the warm-blooded animals in reptiles, fish, amphibia, you have a much lower rate of energy use in the brain. The prediction that follows is that their relative brain size should be much, much, much smaller, because you can still provide the brain, a brain of a certain size, let's say, with energy, even if the body gets very, very, very large, right? Which qualitatively seems to be exactly what you find in nature. Quantitatively, we need data on metabolic rate in a number of species, and there's very little out there.
Thank you.
Well, thank you for the talk. And what about invertebrates? I mean, many of them have open circulatory systems, so you don't have to worry about capillaries at all.
I have no idea. But now there's predictions that you can make and see how they apply.
Many of them can do very, very fine with very, very few neurons.
Oh, I have no doubt about that. I don't think like my thing about animals do what they can with what they get. We know that life doesn't need neurons to begin with. Nobody needs a brain to be alive. Only animals, by definition, are the creatures that have a brain. And even then, you don't need, if you're an animal, you don't need lots of neurons to just go about your business. Once you do have more neurons, then there are a number of consequences, like, for instance, the predicted limit to how large your body can grow around that brain that you have. But I have no doubt whatsoever that nobody needs a lot of neurons to function. It's just, if you have more neurons, then there's a number of things that you get along with that. Like I said, more cognitive capabilities and a longer life, so more opportunities, more time, and as opportunities to use those neurons.
Yeah, very nice talk, appreciate it. So I don't really have necessarily a well-grounded question. It's really a consideration here. Okay, so I'm interpreting that there's this upper limit as to the amount of energy a brain can use relative to body mass and so on. Okay, our experience is people have different degrees of intelligence. Pathology impacts efficiencies of the brain in various ways. So is our attention perhaps then some of the root cause of pathologies and perhaps variance in cognitive abilities directed to the efficiency of the brain? So the brain does lots of things. Cellular metabolism is engaged in all sorts of activities. But perhaps what activities, what kinds of neurotransmitters and so on are being impacted given the available supply, that actually might be a key determinant as to how effective one brain is versus another. Is that kind of necessarily a logical distillation of your understanding?
Yes, but I would actually start with something even simpler than that, which is it's one thing what brain you have with how many neurons you get to start life. It's something else entirely what you do with the brain that you got, which again is where education comes in and opportunities to train your brain and hone and shape your networks through experience. But yes, to speak to your question about pathologies, of course, there are any number of modifications that will impact, some will impact the brain as a whole, all networks at the same time. Others might impact one network and not others. Then you have also modifications throughout life that come with aging. We know now that with aging, you have a decrease in the rate of energy supply to the brain. It happens very slowly, so presumably, well, evidently, the brain has the opportunity to adapt and adjust to those changes, but they are changes of a magnitude that if they happened instantaneously like that, anybody would lose consciousness. So the consequence there is that, well, yes, the aging brain that does lose its energy supply progressively, it cannot function in the same way. Notice that I did not say as well as. I'm just saying it cannot function in the same way as a younger brain that has a higher rate of energy supply.
Well, just another comment if I could, and this perhaps comes out of left field and not necessarily relevant, but here we go. So the brain is a complex system. Physicists have been studying complex systems for a long time. One name in particular is Ilya Prigogine, who got the Nobel Prize in 1977 for his studies on non-equilibrium systems. Okay, so what we learned from that was that any complex system will spontaneously adapt to maximize entropy throughput. That's the fundamental property of physical systems. And I'm just wondering if there's any consideration about that perhaps hard limit that might
Yeah.
reflect on some of the findings that you're getting.
So this concept of optimization, I believe one should only use that word when there are alternatives. Something cannot be said to be optimized or optimal for something if there was no other possible configuration for that system. If there are no other possible configurations for a system, then whatever configuration it has, doesn't matter how good or bad, efficient or inefficient it seems to have, it is the configuration that is dictated by the laws of physics. In this case here, what I've shown you is a very simple equation that captures 65% of diversity across many orders of magnitude, which frankly in biological terms, it's pretty much unheard of. Biologists are usually, for reference, biologists are usually happy with R squares of 0.2, like you explained, 20% of variation in a system. And so far, there are no outliers there. Definitely the human species is not an outlier. So if there are no outliers, if it turns out, let's say, that every single data point does conform to this prediction, that this prediction does work universally, then I believe that that cannot be used as evidence that every single animal is optimized, that you cannot say that body size is optimized to feed the brain. No, body size is the maximum body size that it's the size that was attained. It's the size at which no further growth was possible because all the energy was already being diverted to the brain and supporting the body. So I would say that that is a very complex system, yes, but no, you cannot say that that system is optimized. That system is just the result of the, let's say, of following the law, abiding by the laws of physics.
Okay, so that we can get to our restaurant more or less on time, we're going to take one final question here.
Thank you for the very interesting talk. Throughout the talk, you were talking about the size of the brain versus the size of the body, right? Mm-hmm. So my question is, what about the different regions within the brain? Say, for instance, cortex versus rest of the brain. Because in species, we see that the cortex is bigger in some species. So what are your thoughts about different regions within the brain?
So those can be predicted as consequences of relative allocation of progenitor cells to different parts of the brain, like what's the ratio between progenitor cells that will form cortex versus progenitor cells that will form the cerebellum. There are very interesting scaling relationships that apply across wide varieties of species, although there are clade-specific features like those that define primates. I believe that in the end, put all of those together and you will still get a universal relationship given all these, let's say, the idiosyncrasies of different types of brains and how many. So I can tell you, for instance, that this equation describes what has puzzled my colleagues for the longest time, which is how come primates have a relatively small body, which is presumably, well, that's the size of the sign of the expansion of the brain, and well, it's exactly what you would predict if you had a mutation that just like that doubled the number of neurons that you have in the cortex, which we know that is a feature of the primate brain. You have higher densities of neurons. With a higher density of neurons in the same size cortex, what you have is a higher rate of energy use per gram of tissue. Consider that as the body grows, it provides the brain with less and less energy per unit gram, and you have an animal whose body will be done growing much sooner than in a non-primate animal. right? So the large relative size of the primate brain and also of the songbird brain, of the parrot brain, can be explained as a direct, inevitable consequence of that increase in the number of neurons, in neuronal density, essentially, in the brain.
We've got to wrap it up. So let's thank Dr. Herculano-Houzel again.
Thank you.